In this topic, we'll be introducing what we call classical or Mendelian genetics, looking at how traits are inherited and passed on from parents to offspring, and the relationships between these different characteristics and how they show up or do not show up within a certain individual. So first of all, we have to talk about the very basics. So DNA is short for deoxyribonucleic acid, and this is the molecule that contains all the instructions for life. All of our cells have their instructions for how to make proteins present within the DNA. And actually, every cell within an organism, with a few exceptions, has the exact same set of DNA, the same set of instructions. One thing you'll notice over here in this picture is that not all of the DNA contains these instructions. Certain parts do, which we call genes, we'll talk about in a little bit, other parts don't. So the genes contain the code. Each gene contains a code for a single protein. So from one gene, you can make one type of protein. And there's a certain code that's unique for each different protein that we might make. Here's a short video to introduce the founder of genetics and some of the terminology that's associated with these sort of basic foundations of classical genetics.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett Square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers his lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett Square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited. But it all started with Mendel and his peas.
So let's talk about these terms. First of all, there's a new term to introduce, and that is genome. The genome describes a single organism's complete collection of DNA. Every species has a unique genome, so you may have heard of the Human Genome Project. What that's describing is the genetic code, the collection of all of the DNA that makes us human. Now, part of this is the genes, the part that codes for proteins, and then part of it's this non-coding DNA, which we'll talk about later. So the genome is a complete collection of DNA. We learned about in lab how the genome is broken up into smaller pieces, and each of those smaller pieces is wrapped around and associated with proteins into structures called chromosomes. And then regions of those chromosomes, or regions of that DNA, is where we have genes. The specific code then, each gene codes for one specific protein. This term here, locus, is just the location, so the location on this chromosome here, for the flower color gene, the gene that codes for flower color. This word here, homologous, to remind us, means two of the same types of chromosomes, so two copies of the same type. So if a gene is the sequence that codes for the production of the protein, everyone has the same genes. So all humans actually have the same genes. Despite what you may have heard, we do not have different genes. We all have the same genes. For example, we all have a gene that controls the shape of our chin, whether our chin is smooth and rounded or has a little cleft, a little indentation in the center. So even though we have all the gene that controls that, we have different versions, different variations of that gene. And the term that describes that is allele. A lot of times students have difficulty pronouncing this. It's pronounced like this. Allele. An allele is a variation of a gene. So at this same location, one person might have this sequence, A, T, T, G, C, G, etc. That's the code that codes for the protein that regulates chin shape in this example. These two sequences, these two different versions here, two different alleles, are almost identical. The only different parts are the parts that are in blue and red. So there's slight variations that then end up coding for different proteins that might give you different end results. And we'll talk about what terms used to describe that physical effect that you can sometimes see, a smooth chin or a chin with an indentation called a cleft chin. So I've mentioned that all of our cells, with a few exceptions, have the same DNA inside of them, the exact same sequence, the same code. So how is it then that we end up with different cells, like red blood cells, white blood cells, liver cells, etc.? Well, cells go through a process of specialization, which is what we'll talk about, how that happens. How do you go from these precursor stem cells that have not yet chosen a path or a fate to become then different types of cells? So we'll watch a short video here that's going to talk about this process of gene regulation that actually is what makes cells different from one another.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous, like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be.
So let's stop that video there. It gets to be a little bit more complex than we need to get into. And so as the video mentioned, the reason that cells are different is not because they have different DNA, but because different parts of that DNA are being used. Different genes are being turned on or turned off like light switches. And each cell actually can turn on and off different light switches in different stages of development or different times of day or different conditions, like when you're sick, different genes are going to be turned on and turned off. And so in this example here, in this picture on the right, we have 4 different types of genes. Housekeeping gene that's present and active in all genes, keeping the cells kind of running and normally functioning. Darkened in means that cell is active. We have hemoglobin, insulin, and myosin, genes that code for 3 different types of proteins. Now, red blood cells, we know that they're filled with hemoglobin, the protein that helps to transport oxygen. But muscle and pancreas cells do not make that protein, even though their DNA has the code for that. Similarly, red blood cells, before they mature and get rid of their DNA, they have the code for how to make the insulin protein, but they don't make it. Neither does muscle cell, only pancreas cells do. And myosin we haven't talked about, but it's a protein involved in contraction, leading to movement or work. So muscles have myosin protein in them. That gene is active, but it's not being used in these other ones. As I'd mentioned over here, changes in which genes are on or off can occur based on different conditions, exposure to things in the environment, internal signals like hormones, growth, development, etc. And so we said alleles are different versions of genes. Well, alleles, in part, are determining then those characteristics, those visible traits of individuals. In this example here, these different alleles coding for different colors of flowers are then controlling that trait. Remember that in human beings, alleles are present as pairs in our body cells. So not our sex cells. Our sex cells only have 1 copy of each. But in our body cells, because we get 1 copy from dad and 1 copy from mom, we have 2 alleles for every gene. So a pair of alleles for each gene. This is now going to get into these 2 terms that were introduced in the video, genotype versus phenotype. So because we have pairs of chromosomes in every body cell, that means we get 2 alleles for each of those genes, as I already mentioned. So the genotype is which combination of alleles do you get. In this example here, you can have 2 W's, 2 B's, or a B and a W, each letter representing one of those different alleles. Excuse me. Phenotype then is the observable characteristics. As I mentioned earlier, this is partially determined by the genotype, but it's also influenced by the environment. And we'll talk later about some examples as to how the environment can influence the phenotype. So for example, let's say that you have some corn, and the genetics of that corn allow it to grow to be 6 feet tall. So it can grow up to 6 feet tall. Well, that's great. But if that corn plant doesn't get enough water, or enough sunlight, or enough nutrients from the soil, it's not going to grow to be 6 feet tall, right? Even though the genetics have that possibility, without the right environmental conditions, that phenotype of 6 feet tall will not be achieved. So the environment plays a role in the phenotype. Other examples. Probably most of us are familiar with the pink flamingo. The pink flamingo is pink because its genetics allow it to take a pink pigment from its food and then use that to make these colored feathers. But the pink flamingo cannot make the pigment on its own. So if it doesn't get this proper food that it needs to eat, it can't make that pigment by itself. So that's a case where the environment, in this case the food supply, is affecting the phenotype of that pink flamingo. And the last example of environment affecting the phenotype. In our area, here in the Washington, D.C., greater metropolitan area, the snowshoe hare does not exist. Its habitat does not come down this far. But where I grew up in northern Minnesota, where you get snow on the ground usually in October or November at the latest, and it doesn't melt until maybe April or in some extreme cases May. So you have a white covered area for over half of the year. If you're a rabbit, snowshoe hare, during the winter time you do not want to have this dark coat because you're going to stand out. Very, very easy for predators to see you. And so what happens is as the hours of daylight decrease and the temperatures decrease, different genes get turned on and other ones get turned off to cause that snowshoe hare to shed its summer fur and grow this white winter coat. So it helps with camouflaging. So that's another example. If you took the snowshoe hare and then put it in a zoo, say in Florida, it would never change color because the environmental cues would not be there to cause those genes to turn on or turn off. So let's say here we've got two homologous chromosomes, so two of the same chromosome types, and they have two alleles coding both for purple flowers. So then obviously they're going to make purple flowers. Well, now if you have a plant with two alleles for white flowers, well, obviously they're going to make white flowers. Hopefully that's clear. But the question is then, what happens if they have one allele coding for purple flowers and the other allele is coding for white flowers? What color flowers would the plant produce? How is that determined? Well, that depends upon the type of relationship that exists between those alleles. What type of dominance relationship is there? And that's what we'll be talking about here in the next several slides. So the first example of dominance we'll talk about is called complete dominance, where one of the two alleles we say is completely dominant compared to the other allele. The other allele is considered to be recessive. So what does that mean, completely dominant versus recessive? Well, any allele that is considered to be dominant, it means that its effects are always going to show up in the phenotype. It does not matter how many copies you have, one or two, its effects will always show up. So dominant can't hide itself. Recessive alleles, on the other hand, are the opposite. They're always going to be hidden in the phenotype, unless that's the only copy of those alleles you have. Because if you have a dominant one with the recessive, the dominant will hide that recessive one and it won't show up. So recessive must be only recessive there to have them show their effect. So for dominant alleles, we use a capital letter. And then for recessive alleles, we use a small letter. Easy way to think about this is if you have a big letter and a small letter, the small letter won't be able to hide that big letter, right? But the big letter, that could hide the small letter. And so you might be able to see that small letter, that's that big letter, that dominant one, is hiding the effect of the small. But the small can't hide the big behind it. So let's talk about how we can apply this information. So this is something that we go through in lab. And it's looking at how we can taste different types of different tastes. looking specifically at these bitter taste receptors. Okay? And we're not going to get into the details over here on the right with the physiology of it. We're going to talk about the genetics of it. So there's a certain chemical called PTC. That's short for phenylthiocarbamide. You do not need to know those details. It's located on chromosome number 7, and even that's not important. What is important is a gene that controls the ability to taste this harmless chemical is found on an autosome. Autosomes are chromosomes number 1 through 22. So we call that an autosomal trait. If the gene is on a chromosome number 1 through 22, call it an autosomal trait. Because it's an autosomal trait, it doesn't matter whether you're male or female. Everyone gets 2 copies of these traits because everybody gets 2 copies of chromosomes 1 through 22. So 2 copies of chromosomes 1 through 22 plus either an XX or an XY, which ends up with 46 chromosomes in our body cells. So everyone gets 2 copies of an autosomal trait, 2 alleles. Why? Well, remember, one allele always is coming from dad, and the other allele has to come from mom. It's always 1 from each parent. You can't have 2 from one parent and 0 from the other. The ability to taste this harmless chemical, PTC, is controlled by a dominant allele. So we call it a dominant trait. Because it's on an autosome, we say it's an autosomal dominant trait. I have a few additional terms here to introduce. Homozygous versus heterozygous. The prefix homo means same or similar. Hetero means different. So if you have a pair of alleles and you say they're homozygous, that means they're both the same. So you have 2 dominant letters here, homozygous, dominant. 2 recessive letters are the same ones, homozygous, recessive. Now 2 different ones, dominant and a recessive, they're different alleles. We say those are heterozygous. These terms are going to come up again and again in lecture and in lab, so it's very important that you get these down. Here's an example. We're looking at 3 different genes. So gene loci, the locuses, loci is plural for a locus, 3 different genes. 1 there, 1 there, 1 there. And each of these then are alleles on these 2 homologous chromosomes, 2 of the same type of chromosome. So genotype, the combination of alleles. Here we have 2 of the capital P's, here we have 2 of the small a's, and here we have a big and a small. So homozygous dominant for the P allele, homozygous recessive for the A allele, and heterozygous for the B allele. I'm going to have this posted under the folder for this topic, and I encourage you to do this for practice, Cooperative Activity 4-4 in the Human Biology Condensed book, if you have purchased that. All right, that finishes up this topic.